he said, I think it's almost as important as attending this meeting. So I feel pretty lucky, because I will have the chance to talk to this audience. But I just hope I know what I'm talking about. So what I'm talking about, the sequential gene targeting in the cattle. Looks like this title does not fit today's talk very well, because everybody talk about eggs, ESLs. But you found that we totally depend on the egg, the power from the egg, to allow us to do this work. So if I had the chance, I would add a subtitle saying, many thanks to the eggs. So why we want to do gene targeting in the cattle, in the cow? That's because at Hematech, we have a mission. We want to generate a type of a cow which can produce pure human antibodies. So in order to do that, we not only need to insert human antibody genes into the cow, but also we need to knock out all the bovine antibody genes. And then in order to reduce the concern of medical disease, we also want to wipe out all the prion gene in the cow. So we have achieved all of these. Right now we are formulating vaccines and immunizing these kind of special cows. By the way, the cow were made by nuclear transfer from the genetically modified cells. And we are collecting, purifying, and the bulk product, which we call active pharmaceutical ingredient. So hopefully we are going to make a lot of money. But in the cattle, to do gene targeting, even a single run is difficult, not mentioning sequential targeting. For example, compared to the mouse, we have ESLs. These cells are immortal, divide rapidly, and have high homologous recombination rate, which are the major essential component for anyone to do genetic modification. And because mouse has very short gestation time, you can achieve homozygotes by typical breeding. But in large domestic animals, this is impossible. For example, in the pig and the sheep field, ESLs are not available. So fibroblast cells have been used for this purpose. Because we know that in the primary cells, the homologous recombination rate is much, much lower than the ESLs. So what the research in these two fields did was that they used a promoter-less construct strategy. However, such strategy requires the gene, whatever gene you want to target, to be active in that type of cell. If the gene is not active, this strategy won't work. And again, because pig, sheep have much longer gestation time, and if you want to produce homozygous genotype by breeding, it's very difficult. And in the bovine field, we have an even more difficult situation. For example, especially at Hematech, because we have a special goal of deleting the immunoglobulin genes, because these genes are not active in the fibroblast cells, which are the cell type we use for cloning. So the promoter-less strategy, which can be used in other genes, won't work here. And the selection by the activation of the second allele won't work, which I forgot to mention in the sheep and the pig. What they did after they target the first allele, they use enzymatic selection, just wait for spontaneous mutation for the second allele. So that's how they achieve the homologous targeting of a gene. And in the bovine, the gestation time is 9 months. So we made an estimation from the time you knock out the gene in the cell and to the time you produce homozygous knockout cows, it takes about 6 years. So it's very difficult. If you want to do sequential targeting, meaning you want to knock out another gene, it takes another 6 years. So it takes many, many years. So we figured that if we really want to achieve our goal, we have to figure out a new way to do it. So this is the way we develop. We first transfect our knockout construct to the fetal fibroblast cell. This is a bovine cell. And then we do whatever required to characterize these cells to see if these cells are appropriately targeted or not. But after we finish this, these cells can senesced, or close to be senesced. If we want to do another round of targeting, it was impossible. So we use the egg to do some trick. We put the genetically targeted cells into an egg to do cloning. And the egg, rejuvenate the cells, make fetus. And then we derive cell lines from these fetus. And so we just keep this cycle forever. Seems to me right now there are no limitations yet. We can do many, many runs. So the egg here just like a power generator, each run. So once we achieve the desired genotype, embryos will allow them to develop to term, make calves. So this is strategy. And this exactly what we did for the knocking out of the first gene, which is the bovine heavy chain mu chain heavy chain gene. So what we did was that we sequenced the locus of the IgM, and then found the polymorphic sequence, so to distinguish these two alleles. And then we build a knockout vector. This vector, we have positive selection. Here we use a pure gene, which is flanked by log P sequence. I'm going to talk about why we need a log P sequence here. We also have the negative selection, and then we put the transcriptional and the translational stop cassette in this knockout construct and insert into the exon 2. So we build this construct, we transfect this construct into bovine fibrillar cells, and we obtained 446 antibiotic resistant colonies, and 2 of them, see the targeting rate is very low. And if you have experience doing targeting in mouse EI cells, you know this is unacceptable. But for us, we were pretty happy. And by sequencing the knockout allele, we found that one well contains A knockout, another is B knockout. Pretty good. So then we use the strategy I just mentioned, because after we characterize these cells, we know they are appropriate targeted, but they are kind of senesced. So what we did was that we did the first round of nuclear transfer. So we call it generation 1. We totally transferred the embryo to 30 recipients. At day 40 to 45, we get about 50% pregnancy. And we retrieved totally 6 fetus from these. 3 turn out correctly targeted fetus. 2 fetus contains the A allele, which was targeted. 1 fetus contained the B allele, which was targeted. And then we use these cells, we produce calves to another round cloning. So we call it G2. So the efficiency is close to the primary cell lines. We produce 13 very healthy calves. And this is just the characterization of these fetus and calves. This is the PCR, genomic PCR. We use one primary inside of knockout vector. Another one is external. So wherever you have a correct knockout, you see the band. And this the real calf we clone. This is not a picture we steal from somewhere. This is real clone we clone. They are homozygous in the IgM locus. So we were pretty happy, and then we felt we can knock out the second allele. So for the second allele, what we did only just replace the selection marker in the knockout vector. Same vector, we transfect into the already heterozygous cell line. But you can imagine there are 2 events may happen. One possibility is that this targeting vector will target the wild type allele. So in that case, you have a knockout. And then because another allele already knocked out, you will have homozygous. Another possibility is that because the same vector may replace the already knockout allele. So in that case, you still end up with the same allele knockout. But we went ahead, did the experiment anyway, because we have 2 types of cells from the first round targeting, A allele heterozygous, B allele heterozygous. So when we transfect this construct into the A allele heterozygous cells, we screen about 1,200 wells, 5 of them turned out to be correctly targeted, and 2 of them targeted B. So that means we generate homozygous BOVINE fibrillar cells for that gene. But 3 targeted to the A allele. So to replace the original knockout, which is not what we want. But it's okay, we have what we want. In the B allele, the similar thing happened. We screen this many numbers. We have targeting, we have replacement. And overall, if you look at A allele, see you have 6 cell lines targeted in the A allele here and 3 here. So totally we have 9. But B allele, we have 1 here, 2 here. So overall, the preference, somehow the vector like to target the A allele a little bit better. So then we use these cell lines, homozygous cell lines, did another run nuclear transfer. So we call it G3 clones. And then we totally transferred to 83 recipients. At the day 45, we got 45% pregnancy, and we recovered 20 fetus. And 8 of them are homozygous. And then we use these cell lines to do another round cloning. And we transferred to 137 recipients, 8 calves were born. And the efficiency, if you remember, the first round is about 8%. We do not feel it's significantly different. So 4th round cloning, we still feel we have an efficient way to clone these cells. And this is just the characterization of the fetus. We do PCR again. If you have homozygous knockout 2 alleles, you see the band. If heterozygous, only 1 show up. We also did a negative PCR. And by the way, we also sequence these locus. We totally convinced we have the homozygous knockout. And then because at day 90, the IgM gene was expressed, so we did RT-PCR. This is just the controls. This is homozygous knockout, so the gene was totally knocked out. And then these are the calves. Again, they are real calves, and they are homozygous. So these calves are about a year old right now, actually. So we were so encouraged. So we want to knock out the prion genes, and we feel ready. But before I show you the work, I want to draw your attention to the limitations of using the positive selection when you do sequential targeting. See, in the 2 round knockout, I just show you, each round you use different selection drugs. So if you want to knock out 4 genes, you need 4 different drugs. In the Hematech, we probably want to knock like a dozen, maybe more than that. So probably you will run into a problem to have enough selection genes. So we use another strategy. We use the selection marker removal strategy, which is explained here. So that's why we put the LOXP site here. Because in these homozygous knockout cell lines, if we transfect the cell line by CRE gene, we'll delete these positive select genes. So these genes can be used again. So we basically recycled these genes. And this exactly was done. So we did that. We transfect CRE gene into the homozygous cell lines. We characterize the cell line. But then these cells are kind of senesced. So we did another round of cloning to rejuvenate the cells. And we transferred 60 recipients. We got 21 fetus at a pregnancy rate at the day 40, and we recovered 5 fetus. And turn out that all of them have the CRE successfully loop out the 2 markers. But even we use a minimum drug selection, use a circular CRE expression construct, 4 of the fetus have the CRE gene integrated, which is not what we want, because we do not want the CRE gene there. Because if you have the CRE gene there, the second run, the following run, before the gene targeting takes place, the selection cassette will be looped out by the CRE gene. So, but luckily we have 1 cell line which does not contain integrate the CRE gene. So we use that cell line for the PRP knockout, prion knockout. Again, this is just the characterization after the loop out. When we do PCR, you have small band, but before CRE transfection, the pure will generate 2.4 kb, neogene will generate 4.3 kb. So we confirm this. And then we were ready to do the prion gene knockout. We used a similar strategy, positive selection, negative selection, and here we use neo because neo can be reused again. So I just simply call these two alleles C allele and D allele. We transfect the construct into the IgM homozygous knockout cell line. And we screened 203 antibiotic-resistant wells. 13 of them contain the correctly targeted cells. But very interestingly, all of the wells, 13 of them, all targeted to C allele. No one targeted to the D allele. Although there are only one nucleotide different, we don't know why that was the case. Some people believe maybe prion gene 2 allele, one is active, more active than another. The active one may be preferably knocked out. And another observation was that we found about 10 times higher homologous recombination rate in the prion locus than in the IgM locus. And again, we believe that because the IgM gene is not active in fibroblast cells, but the prion gene is active. So active gene probably will give you a more open chromatin structure to let the knockout vector to do homologous recombination. And then after we characterize the cells, we did the same trick. We went ahead, do another round of cloning. So now it's G4. We totally transferred to 39 recipients. We got 28 pregnant at day 40 to 45. And from those, we recovered 5 fetuses. And all of them turn out, all these fetuses are IgM homozygous and PRP heterozygous. So we basically have a triple knockout already. And as I mentioned, in this case, all the C allele was targeted. So this is just the characterization of these cell lines, the fetus. We do PCR, negative PCR, positive PCR, and we were convinced they are triple knockout. And then we had a little bit problem because, first round, as I show you that all the all the cells we identified was targeted to the C allele, not the D allele. So we were thinking, will the same vector, after you replace, you change the pure positive select gene targeted to the D allele or not? But we gave it a try. So turn out, we obtained 327 antibiotic-resistant wells. 17 of them is pretty high efficiency, well contains correctly targeted cell. And this time, the cell somehow knows what we want. They target, most time the D allele was targeted. And we cannot explain this very well, but if you can imagine, to target D allele and to replace C allele is a little bit different situation because here is a replacement because you have the constructs, the stop codons, so the DNA configuration is a little bit different. So, but nevertheless, we like this result a lot. And then we went ahead because in order to produce CALVES, you need to rejuvenate these cells. So we did another round of cloning, so they called G6. And we transferred the embryo to 67 recipients. 46 of them were pregnant at day 40. We recovered 18 fetuses, and all of them are quadruple knockout, full gene knockout in the fetus. And this is just the PCR characterization of these fetus. Again, this is the prion locus. We check it is homozygous And also we did a negative PCR and turned out it's negative. And then we look at the prion gene expression in the fetus by RT-PCR and turned out they're negative. So we truly achieved the quadruple knockout and 2 genes sequentially. And this is just a short summary. So we achieved that gene targeting both in silent locus, also active locus. Although the efficiency is kind of different, but we can do it. And we demonstrate that targeting vector replacement is a good strategy. If you flunk the positive select gene by LOX P site, you can reuse the positive select marker again and again. And we demonstrate that we can clone at least cow many generations. And because we, each time rejuvenate the cell and do not need to wait the fetus develop to term. At day 40, we recover it. And the generation time is much shorter. So on average, about 2.5 months, we did a run targeting. And because we are so proud of our work, and I would like to share these comments from Dr. Yang, who is from University of Connecticut. He was supposed to be here. But what he wrote was that he said, nuclear transfer cloning touched off a revolution in genetics. A new study by Hematech, I add this term. I know what he means. Outlining how to target genes serially in cow ushers in a new era in large animal genetics. And one sentence I would like to add is that we not only can publish this paper and demonstrate on paper is feasible, but we also already industrialize this method. We are not many more genes, but because the confidentiality reasons, I'm working in the industry right now, and I cannot see much. But we can clone calves at G6, maybe more. Yeah. And sorry, a little bit of acknowledgement. Yeah, this project is directed by Dr. Robo, who is the president of Hematech, and also Dr. Asol, who is the senior scientist from the pharmaceutical division of Kirin Brewer Company. And Dr. Kurowa is the director of the molecular genetics, by whose team the genetic targeting was done. And Dr. Kasinathan is the director of the cloning group. And his group did all the genetic nuclear cloning. And as true for any experiment, without these small people contribution, nothing can be done. So all the people from this department contribute this work. Thank you.
Well, thank you. I look forward to drinking a glass of beer and being fortified against influenza. Carol?
You're a company and you want to make money, and it seems to me if you could knock out all the prion genes, that might be economically much more favorable to just make herds of cows as a source of meat than a guaranteed prion-free. And forget about the antibodies altogether.
Right. That's just the backup. Yeah, we don't think that'll make us rich enough.
So you've thought of that already.
We thought that.
Yes.
Healthy hamburger.
What is the story with the large calf syndrome? I mean, you guys seem to be producing a lot of calves. Is that problem gone?
Problem not gone. I show you 80%, 10%. That means 90% still have problem there.
So, but a lot of them are dying at birth, or?
That's right, before birth even. That's right. Once you see the calf, C-section, the calf come out, want to jump, that calf will survive and won't have any problem. So most of them die during gestation, and some of them, born like, the egg cannot extend, cannot stand up. We try to rescue them. We take very good care of them, but they last like a month or two.
You lose them.
That's right. Yeah.
But that abnormality is obviously due to some epigenetic change due to the nuclear transfer cloning. Right. Now, the fetuses that you take, if you take them late in gestation, then many of them will have already died,
That's right.
so you will have selected against the epigenetic changes. But if you take them early, there's perhaps less time for epigenetic changes. What stage of gestation do you take the fetuses?
40 to 45 days.
40 to 45
That's right.
days.
Actually, you are absolutely right. We would love to wait until the healthy calf born and then take these cells because they have gone through all the selections. However, they take much longer time. So we take the risk. We instead retrieve only one fetus, we retrieve a bunch. We derive cell lines. We try them simultaneously.
Nice.
Some of them, I have to tell you, never give us another round of fetus. They do not work at all, but others work.
So when you're testing them then, you test them only for when they work or not. You don't look at the genes and say, oh, there's a change there. Oh, there's a change
We
there.
have a basic research program going on. Actually, this Monday, I'm going to organize a collaboration. We have two collaborators from other universities. They have been looking for the placenta gene expression in this network.
Yeah.
We look at the epiblast lineage. So a lot going on there.
Yeah. Very nice.
Tell me, when you do the knock-in experiment, do you see multiple sites of integration?
No, actually, because we have next selection that is reduced. But indeed, we have drug resistant colony. When I show you like 100, but only like few of them correct target. And these cell probably you have other site integration. Maybe you have multiple, but we have not paid too much attention to this. We just care, okay, if this is the case. Also, we do the negative PCR. If you have a correct targeting event, but at the same time have another knock-in, we do not want that. We have a scheme to distinguish these two.
So you're screening those pieces with PCR so right after the fusion, after the selection.
That's right.
Okay.
Tell me in a 5-minute break.
We have a 5-minute coffee break.